In this video, we will continue our discussion about how this T-cell immunotherapy recruit macrophages. So, in the previous video, we have understood the abstract about this paper. So, in this video, we will start discussing the results that are presented in their main figures. So, in order to check and validate whether T-cell immunotherapy recruit macrophages and other immune cells, what did it is they did this T-cell immunotherapy experiments on two different models. First one is this RMA QA1B engraftment in the mouse, and second one is this B16F10 engraftment on the mouse. And then in the first case, they inject the vaccine or the T cells with the receptors to target the tumors at 12 days, and in the second case they inject the vaccines and simultaneously they also inject this bispecific antibodies to increase the immunotherapy impact. So we will start discussing from the panel A. So here they took the mice model and inject this engraftment, that is RMA QA1B negative, and this is the standard model to check this T-cell immunotherapies. And then after 12 days they inject the vaccines and then observe the tumor weight and whether the mice will be able to survive or death. And what they have find out is that compared to the untreated case, there are several mice in which the tumors are suppressed, and in some cases tumors are eliminated completely. And thereafter they wanted to check what are the changes that occurs at transcription level between the treated cells and the treated mice models and the untreated mice models. So in order to compare that, they differentiate, since some of the mice models that are affected with these vaccines are also does not show any response in terms of the increase in the survival rate. So in order to distinguish between those two samples, what they did is that they distinguish the patients, the mice that are treated with the vaccine into two different categories. First one is non-responder and second one is responder. So how they define the responder? They define the responder is by that if after this injection of the vaccines, they on the 12th day and on the 19th day from the day start of the experiments, what they did is they check whether the mice has this tumor regression symptoms or their symptoms does not show any tumor regression. And if there is a tumor regression, they consider them as the responder, and if the mice does not have any tumor regression, then they consider them as non-responder. And when they check the various immune cells in those at transcription level in these three different categories, there is untreated and non-responder and responder, they find out that in the case of responder there is a increase in the number of the immune cells such as macrophages, CD45 cells, neutrophil cells, CD8 plus T cells, whereas in the gene expression levels at the non-responder is more or less similar to the untreated. So this indicates that the responder cells or the mice that are responded with the vaccines has high immune infiltration, high immune cells infiltration at the site of tumors. And in order to further check their relationship with the T cells, what they did is they separate out the populations in this in the mice, in the responder mice, and do the flow cytometry analysis, and from that they are able to find out the relationship between these T cells with the macrophages and NK cells. They find out the relationship between these CDT plus cells and NK cells, macrophages, neutrophils, and in all the four cases what they are able to see is that they are able to see there is a increase in the infiltration of the CD8 plus T cells, NK cells, and also these macrophages and neutrophils in the responders. That is, they have a positive relations. And within the macrophages and neutrophils, they wanted to check what kind of their subset populations are shows a positive correlations. So when they check the M1 type macrophages, they see its positive correlations, whereas in the case of M2 type macrophages they see its negative correlations. And similar correlations they find out for the NK type neutrophils and N2 type neutrophils. That is NK type and N1 type neutrophils has a positive correlations, whereas N2 type neutrophils has a negative correlations. So after validating their uh, positive correlations between T cells immunotherapy and the immune cells recruitment at the site of tumor, they wanted to make sure that this is reproducible, and this phenomena is reproducible. So what they did is they did the experiment in different tumors and different mice models also, that is B16F10 engraftment. So in this case they not only use the vaccinations but also use the bispecific antibody injections to increase the effect of uh, treatment. So in this case also they find out that in in the cases where mice are treated with the vaccines and bispecific antibody, they see an increase in the survival rate, or they see a delayed tumor progression. And when they check the immune cells in these two different populations, they are able to find out that in the case of these untreated cells, there are less number of immune cells are infiltrated at the site of tumors, whereas in the case of mice that is treated with these vaccines and bispecific antibodies, they see increased recruitment of the immune cells at the site of tumors. And when they further analyze what are the percentage of the cells, live cells, in these two different categories, they are able to see that in the case of vaccines, in the case of mice that are treated with the vaccine and bispecific antibodies, they see the increase in the CD8 plus T cells, the NK cells, and at the same the macrophages, these M1 type macrophages that can be seen here. Whereas in the case of untreated cells, what they see that there is a not much increase in the CD8 plus T cells, NK cells, and even the macrophages are low. So, and the N1 macrophages is also high in the case where vaccines and bispecific antibodies is used. But in the case of uh, this untreated cells, the monocytes are higher, but in which way they are directed or towards differentiation is clear that they are not have any positive deviations towards M1 type macrophages like this mice that is treated with vaccine and bispecific antibodies has. This two different models, or these two different results of these two different experiments of the different model shows the similar results or similar phenomena, that this immunotherapy recruits immune cells, mainly macrophages towards this combined immunotherapy effect against the tumor cells. So I hope you understood their point that they have made from this figure. If you need to understand more about these figures in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.